first of all, many thanks, Ben, for this very kind introduction and for the chance to talk to you and your colleague. It is a really great pleasure to have the chance to talk to the next generation of the young medics, and especially to talk about such a fascinating and exciting topic as therapeutic and diagnostic applications of synthetic biology are. So let's just rephrase this talk, that in this talk, I'm actually going to talk about a couple of very interesting applications of synthetic biology in diagnostics and therapeutics. And I'll talk a lot about these. And this is an overview talk. I promise that we will not go deep into any kind of complex biology and bioinformatics and systems biology and control theory and machine learning. The purpose of this talk is really to get you excited about the potential of the synthetic biology and its applications in medicine. It is also to make you aware of some of the challenges that synthetic biology and medicine faces, and basically to get you a bit more into engineering and show you how engineering principles can be applied to building new biology. Because I believe that as engineers or as bioengineers and doctors, they have something very close. We aim to solve the fundamental problems, and we want to solve the big problems, like engineering novel biofuels or engineering new therapies or saving a human life in an ICU. So sometimes the solution might be ugly, it might be dirty, but it has to work. And similarly, engineering things have to work. So why synthetic biology and why does it matter for the medicine? I really hope that at the end of this talk, you will be able to better appreciate and find answers to these questions. So let's start. So what is the synthetic biology? Synthetic biology is an engineering discipline that aims to design and build novel biological systems. I really like the expressions of and quotes of two very famous physicists, Theodore Karman and Richard Feynman. So I really like the one that delineates science and engineers, which is scientists discover that world that exists, engineers create a wall that has never been or that never was. And this is, I think, really true. As engineers, we should aim to not only discover and understand, but especially to create, to develop, to build, to construct. And on the other hand, there is not just this building concept, it is also the fact that as engineers, we can better understand the systems, we can better understand if we can build. As Richard Feynman famously said, what I cannot create, I do not understand. So what we could say, if I say perhaps in the future can build a synthetic cell from the scratch, from literally off-the-shelf chemicals and bunch of nucleotides, then it really means that I really understood principles and understood the processes in the cells. And so what is synthetic biology? It is using engineering principles to design and build new biological systems and living organisms. And what are these engineering principles? We'll talk about this a bit later. So first of all, I want to preface this. Sometimes we can hear synthetic biology and genetic engineering, like, are these the same? Are these very different? Like, what is this? So I just want to preface that genetic engineering, basically, its very simple level means I want to transfer a gene from one organism to another to like make some good things, right? Something simple. So something simple yet very useful. Let's imagine the human insulin. Human insulin has been made in bacteria since, I believe, 1980s or something like that. When people took the gene for human insulin, inserted into bacterium, and then started producing insulin and a fermenter, which is quite great because you don't have to go and isolate insulin from pigs, and more people get a high quality insulin, and it's great. But as synthetic biologists, as biological engineers, what we really mean to do is to build novel, sophisticated biological systems with complex functions, right? So I think that this is very important to appreciate. While as a genetic engineer, I can ask, how do I make molecule X in cell Y so that I get more of it? As a synthetic biologist, I should ask, like, how do I get a biosensor, a sensing cell that can sense 4 different chemicals at 4 different levels to produce the desired output at a level I wish. So with that difference cleared, let's get to like introduction to engineering biology. The basics of engineering biology is formed by the DNA. So the DNA really encodes genetic information, as we know. Here is its structure with 4 bases, 4 natural bases, we should say, because obviously bioengineers have gone further and have engineered a couple different bases into it. So now we have like unnatural DNA with like 8 bases or so. So you can like play around, but in its essence, we have 4 bases paired at SAT, adenine, thymine, and guanine, cytosine, and then we have phosphate backbones. And really, the ordering of these bases determines the genetic code, which in turn determines the structure of the polypeptide chain, the primary structure of the polypeptide chain, primary structure of the chain of amino acids, right? And everybody probably knows this central dogma, which is that we have replication, where DNA basically goes to DNA through DNA polymerase, transcription, where DNA is being transcribed by RNA polymerase into RNA, and translation, where RNA is being translated by the molecular machines called ribosomes into the protein. So essentially, at a very central level, we can think of the cell as somehow as the hardware for our software, which is the DNA, which contains the instruction for the proteins that can do all the set of things from having structural properties like membrane channels up to regulatory properties, enzymatic properties, and so on. So, and what really enabled the inception of synthetic biology has been the decreasing cost of DNA sequencing per base. So DNA sequencing is essentially reading out of the genetic code. So if I can basically take a DNA and read out the sequence of the letters of this genetic code, I can perhaps go and elucidate what this genetic code is doing. And what we see on the left is basically the comparison between something called Sanger sequencing and second generation or next generation sequencing, which are kind of 2 typical sequencing methods, which basically the second one allows us to sequence at much higher throughput, and Sanger sequencing is still being used for small fragments of DNA. The point is that over time, our ability to read the DNA has increased significantly, and the cost has decreased very fast. Well, actually faster than the famous Moore's law, as you can see on the picture on the right. So this has been one of the strong enabling things. So we know somehow how the DNA operates on a certain level, and we can read it, which means that we can perhaps elucidate the function of this DNA. And similarly, as the cost of DNA synthesis, as the cost of DNA sequencing has decreased, similarly, the cost of DNA synthesis has decreased. So basically, we are actually able not only to read the DNA, but we are also able to kind of write it or print it out or synthesize. But essentially, and this cost is basically decreasing as well, not basically as fast as the cost of DNA sequencing, but still fast enough. So now, how will we combine it? Like, we can basically we know what the DNA does at a fundamental level. We can read it, so we can elucidate a function somehow, and we can write it or synthesize it, which means that we can somehow write this function down, which is essentially what we want to do in synthetic biology. We want to somehow write our genetic code. We want to give this instructions to the cell, or even like to cell-free extract on what to do and basically what to manufacture and say how to behave. So really the workhorses of synthesis. So how can we think about it? So really let's think about it. So we can basically now elucidate the function of the DNA, right? DNA makes some proteins. We can sequence this DNA, so we can say somehow elucidate its function. We can write the DNA, so we can perhaps go and print out the instructions for manufacturing of these proteins. And now we need to get these proteins into the cells, which we basically can do on something called plasmids. They're circular pieces of DNA that contain all the important things that are required, say, to produce the protein. Of course, apart from the cellular machinery. So they usually include the origin of replication, so the DNA polymerase can bind there and replicate a plasmid. It contains something called restriction sites, which basically are, we could say a marks where something called restriction enzymes can cut and replace the parts of the DNA, or like the genes of the interest, and also contain, say, antibiotic resistance gene or selectable markers, so that we can basically select out bacteria that actually keep this circular piece of DNA. So bacteria are so we can think of plasmids as somehow as USB sticks, or for the old-fashioned fans, CDs or DVDs that can carry our proteins and our DNA into the cell, and that actually, and then basically cellular machinery, cellular polymerases and ribosomes and amino acid tRNA synthetases, like anything you can think of, then goes in and basically transcribes and translates the DNA of the inserted gene, for example, and manufactures it. And you can kind of see that this is something somehow very important, because what we can do is we can have, say, this collection. You can order this kind of plasmid online with a specific, say, gene of interest inserted, and that encodes for a specific protein. Literally order it, get it, and insert it into your cell. And you can order multiple different plasmids. Multiple different plasmids might work in different cells, etc. And then you can make your proteins of interest in different cells. And you can even go and, say, take two parts that are in two different plasmids, cut these plasmids at these restriction sites that somehow mark the places where you can cut, and then you can ligate or connect these parts together. So you can go and make your own construct. So literally you can somehow go pick part 1, part 2, part 3, like piece of software 1, piece of software 2, piece of software 3, put them together and load them into your cellular computer and run them with your cellular hardware. So as I mentioned, what do we do with the plasmids? Cells are actually, when cells are in something called state of competence, cells are able to go and uptake the plasmid, and then basically it can propagate into the cells, into the next generations, and produce your protein of interest over the next generation. This is called transformation in prokaryotic cells like bacteria, and it's called basically transfection in, say, human cells. And you can also do something different. Basically, today we have a lot of tools that enable us not to just have these circular pieces of DNA floating there, but also to basically go and integrate this piece of DNA into the chromosome of the given organism, so it stays there and has basically greater likelihood of propagation to the next generation of the cells. So really, somehow it's very easy, and somehow we can conceptually think about this as about reading DNA to learn about what it does, what proteins it encodes, what functions it has, and writing the DNA, putting it into the cells, this kind of genetic software, and then running it. At the very kind of simplest level, this is one of the things that synthetic biology can do. So, and obviously we need to basically measure. So there are various things how we can measure if, say, our genes of interest are in the cell and what they basically do. And these include colorimetric assays. So we can have something like beta-D galactosidase assay where basically we can get some kind of color output if if our plasmid of interest contains this marker, so we can confirm that. If we put it into this proper medium, we can get this kind of output, which is good for in vitro measurements. And then we can go and have fluorescent proteins, which are like green fluorescent proteins or red fluorescent proteins or white or yellow fluorescent protein that basically just fluoresce, and you can basically have just measure the fluorescent signal, and you can also like insert a bunch of something called barcodes, which allow you to basically sequence your DNA and subsequently to figure out whether your cell has been properly whether your cell properly contains the DNA it should. Okay, so how does this kind of all works? This is something called design build test cycle in synthetic biology, and this moves us towards our attempt to make biology more of an engineering discipline. So we really want to get iterational design, and we really want our biological circuits to work. So ideally, what we want to do is first to design, perhaps in a computer, choose our host organism, choose our pathway, design our genes, design our specific sequence. Then we want to build all of these things. So we can, for example, as I shown before, like cut the different plasmid vectors to like assemble our target plasmid that can get into the cell and produce the desired function. And then we can go and we can basically test whether our circuit or whether our genetic circuit or whether our basically DNA being inserted into the cell performs what we wanted. So, and this process can now be pretty fast. This process, as you can see at the bottom picture, can sometimes takes up to like 4 days, which is quite fast if you think about it. So if you're sitting somewhere in California where it is easy for you to get to some sequencing and synthesis companies, it's very easy to get this kind of fast overturn. And this is very important because let's imagine that you have a computer program that takes couple of weeks to run and to compile. That's not what you really want. We would like it to run in seconds. Well, this is not where we are in biology, but we can run our biological programs in basically still in a couple of days, which is quite great and quite an improvement, especially if you think about how we've progressed over the last 20 years. And today in synthetic biology, in engineering biology, we have a great set of novel tools from automated design tools like Cello, where essentially you can basically go select the logic functions that your cell should perform, meaning that you can come and say like, if substance A, B, C, and D, which are some specific substances that are present, then produce the certain output in the cell. So say produce substance E, only if all 4 of them are present. You kind of throw this into the computer, select the cell, select the host, and this software called Cello will kind of spit out for you the genetic sequence, literally like the DNA that you should go and order, all the plasmids, all the things that you should go and order that you can then essentially order and go and insert into your cells, which is quite great. So, and this is similar to say what electric engineers are often doing. It's not that electric engineers who are building iPhones are going to like single smallest transistors, right? That's not what we want to do in biology either. We have automated recommendations tools. There is a novel research from Radivojevich et al. From 2020, where you can literally like iterate over your experimental essays, and the algorithm kind of tells you that you should increase or decrease amount of this or that protein in your experiments to achieve your target objective on a very abstract level. We also have some much better tech. We've got a liquid handling robots like Opentrons. So, the days of playing with a pipette are still in, but they're somehow going away, especially especially if you really want to go after the precise results, because if I have a robot that can hold not like one pipette or like multi-pipette with 10 tips, but can do 296 viral reactions at the same time, that's much better, and you can get much better precision if you can program something like this, and your statistical samples are going to be much bigger. And similarly, we are having affordable bioreactors. And this is something that my supervisor, Harrison Steele, and my other supervisor, Antonis Papachriustodoulou, along with Rob Habgood, came with, which is something called KeyBio, which is a bioreactor that allows you to maintain the controllable biological reactions with controllable environmental properties, with controllable, say, amount of different chemicals you're putting in. And that's quite important because for a lot of time, we've been, say, growing our bacteria on the agar plates, which is not necessarily what you want to do. And this is much more sophisticated and can be much more better. So this is on kind of very, basics of synthetic biology. However, what I would now like to talk a bit about is that we should talk about engineering principles in biology. Engineering principles in biology. So I just want to take you through this, and in conceptual terms, think about how we make biology easier to engineer, how synthetic biology can exist through the utilization of this engineering principle. And fundamental engineering principles are, I would say, I like to call them 4: abstraction, characterization, standardization, and modularisation. So for abstraction, what is abstraction? So in abstraction, we are essentially moving from DNA code to functional proteins. So let's imagine that really what is our DNA? It's a huge set of letters with ACGI at the beginning, and then it goes like ATG, CG, GD, AAA. Then it gets transcribed into mRNA and gets translated into amino acid chain, and this amino acid chain then essentially performs certain function of the, say, green fluorescent protein. And essentially, we can think of this process as an input, where the input somehow is the DNA, and then there's like some abstract manipulation, and at the end there is the output, which is the fluorescent protein. So, and in this case, what we really want to do is to abstract. So, in my work as a genetic circuit designer, similarly as an iPhone designer is not thinking about individual transistors, I don't want to be thinking about DNA. Like, I don't want to be like designing circuits like PAAT, ACG, ACT, CAAC, ATAA. I want to go like, okay, like give me something that takes this protein as input and takes that protein as output. And then I want to connect a bunch of these things to perform something useful, say detect a dangerous chemical or basically detect a cancer biomarker and produce a cytostatic drug, right? So from DNA, we are getting to parts which are kind of individual parts of these genetic circuits, like promoters, which is the place that you could say where basically the transcription, the DNA, the RNA polymerase can bind, and where transcription is initiated. Then to the devices, where I can essentially take the whole module. So say I can take this promoter, ribosome binding site, coding sequence, terminator, and this basically constitutes one module, which has as input takes a protein that a transcription factor that activates the transcription of mRNA of my coding sequence, and the output is say this mRNA, and then I can have a system where basically this mRNA further interacts with other such a module, other such device, and forms and basically binds it and forms some output, right? Forms basically some output, say activates the translation of other RNA, or rather enables it through the activation of a toehold. So you see, this is basically something what we want to do. We want to abstract from DNA to whole systems. Then what is key is characterization of individual parts. So you can think about this as an engineer thinks, like I want to have a well-characterized screws, I want to have well-characterized bolts, I want to well-characterized beams, etc. So similarly in biology, we want to describe our parts well. So I would somehow like to have a spec sheet or data sheet for each of my parts, each of my genetic parts that kind of says, you know, this is the part, this is what it does. These are the conditions like temperature, pH, salinity, which you can think of where it works. And this is what it takes as the input. This is what it makes as the output. And for this kind of input, to get this kind of output in these or those conditions. And I would like to do this for each part. And this is still somehow difficult to get to because A, the biology is a bit more chaotic. And you know, B, it's much more complex, like measure these individual parts. You can see, you know, these different kind of mutations, and it's not that easy to like make the same DNA part all around the world. It's not basically just as easy as producing a billion or trillion of individual nuts or bolts. But this is where we are trying to get, right? Say with these promoters, which is something, as I mentioned, where RNA polymerase binds and produces the output. So, that's something that is very similar to industry. We want to characterize our things well. Then standardization is the key property for reproducibility. So what you can see on the right is that we have a plasmid, this kind of piece of DNA, which has some certain structure, just brief, which is somehow the antibiotic resistance marker, ChemR. It has basically some kind of a backbone, and it has a prefix, suffix, and the given part. And this is basically how we want all of our plasmids to look like. We want all of our kind of genetic parts to be stored in structure like this, and so that basically everybody can go and easily take things out of the shell and combine them to produce novel things and novel plasmids, right? Combine different things. But to that, we need to have standardized things. It's the same way as say we have the standards and norms for each of the screw or beam that comes down from the assembly line in a factory. And then what is important is modularisation. I somehow attach this with the abstraction, but what we really like to have is the modules. So basically, we can have the input into our system, and then we don't need to really care about what's going on in the system, but we know what output we get for the given input. It's similar as somehow if you say, you know, have your computer, you press the key A and you see A on the screen, but you don't know what to press A. After you pressed A, that this got translated into some kind of machine level instruction before it went back to be displayed at the output of your display, right? You don't care about that. You know that when I press A, I can see A on the display. And this is something we'd like to do in biology. We want to do things like switching, right? So basically, if I apply this amount of chemical, I will get this kind of switch. If we want to have other module that can be kind of oscillators. So say if I want to build a clock, I want to put in this amount of chemical, and I want to have kind of the output with certain frequency and amplitude, and so on. And these are kind of the modules, and this is what we want to see in synthetic biology to build really complex systems. Because as you can see, say with your iPhone is built from individual transistors. But on the other hand, the guy who programs the apps probably doesn't care what basically these transistors are. Yeah. And so how the history of synthetic biology has gone, it's truly remarkable. So like if you really wanted to go back and back and back and back, you could go to days of Jacques Monod and his Nobel Prize discovery awarded lac operon discovery, where he kind of discovered that the protein production can be regulated. And today, I would say around 70 years later, we got to multiplexed eukaryotic therapies. So modification of the cells and engineering of the cells that take multiple therapeutic diagnostic biomarkers as inputs and produce multiple therapeutic outputs. And really the synthetic biology had its advent or like birth in 2000, when the first synthetic genetic circuits or first complex circuits called toggle switch and reproducibility were constructed. And this has really shown that you can do the functionality that hasn't been observed before. And so now we have like all the kinds of different things like new circuit functions, activate repressor oscillations, increase gene regulators and sensors. And now we are even looking at something like synthetic communities. So we're not just engineering individual bacteria. We want to engineer the whole communities. We want to store information in DNA. We want to insulate parts and really build complex things with biology. And before we get, and so for now, we will soon start talking specifically about the medical applications of synthetic biology. But before that, I want to mention a couple of different applications where synthetic biology is important, which is cost-effective production of complex therapeutics, like companies like Ginkgo do, eco-friendly production of clothes that companies like Modern Meadow do, producing hydrogen fuel in bacteria, which Hygiene or Macquarie University does, sustainably produced animal farm that startups like Hoxton Farm do, and sustainable cotton that Gali works on. And I think that this is just like to show the remarkable breadth of usage is of engineered biology, because it's somehow remarkable. If you think of the cell as a factory for protein, these remarkably complex macromolecules, you somehow just give your cell a simple nutrient or a set of simple nutrients like glucose, some salts, and some amino acids. Your cell is going to make something pretty complex out of it. And can you imagine that just like deliver this very simple materials next to your factory, and this factory somehow itself like makes a bunch of very useful things and even multiplies, right? Because this is what cells do, they do replicate. So this is hardly something possible. And this is why engineered biology is really transforming the world and the industry. So, okay, is this like all beautiful and rosy, awesome world? Well, no, like synthetic biology faces a lot of challenges that we're working on overcoming. So first of all, it's complexity of biological systems. Biological systems are incredibly complex, are incredibly nonlinear. You are doctors and you probably know that often we can't predict what will happen with a patient. It is because the human intuition, and our intuition is somehow built in a linear fashion, really fails when faced with complexity of biological systems, where the responses are often nonlinear and things are interconnected, etc. We also face a scarcity of the well-characterized parts in synthetic biology. Basically the issue is that we don't have billions of different parts, billions of different genetic components that would easily be interconnected to like different things and that would be well described. This is not the case. So our toolkit is still somehow limited. Fortunately, there are companies working on it. Then we observe the lack of modularity. Let's imagine a computer. My computer right now is connected the charger to the socket, and I kind of know that this electric current flows from the socket into my computer. Well, in biology, and it flows nowhere else, but in biology, it's very difficult to somehow, in a cell, connect two different things. It's difficult to connect them directly because there will be kind of off-target interactions and basically things being mixed together, just interacting with each other. And this makes it very difficult to like modularly design and predict the behavior of gene circuits. Furthermore, we have limited shared cellular resources. We've got limited capacity of cellular resources. So basically, if we start adding our artificial genetic material into the cell, what it starts doing is it might want to reject it, because like, why would I bother like making other proteins that I don't want to make, that some idiot just stuffed into me, if I can just go after my own business of making things I want for my survival. And similarly, if we add these additional proteins, the cell will have less resources to produce its own resources for growth. So more proteins I add, perhaps the more protein my cell produces, perhaps the worse it grows. So there is some kind of a trade-off there. Then there's inherent stochasticity. So biological systems are not only complex, but they're stochastic and noisy, and often the concentrations of molecules are so small that we can't just go with kind of mean field approximation and looking at the average concentrations. We have to look at individual molecular amounts, and that's what sometimes difficult to work with, because it's not that you have like 10,000 molecules plus minus 100 molecules, that's still fine. If you have 10 molecules, like plus minus 3 molecules, that's quite of a variation. And then we can often see this kind of variation in concentrations, and we have to work with probabilities, and it's just not, and this can often like ruin the functioning of our circuit. And then in biological systems face a challenge that is completely foreign to man-made systems, which is evolution. You can't just imagine having a bridge or having a car that just one day thinks like, I'm really bothered by this screws or by this wheel or by this, whatever light, like I don't like it, I'm going to kick it out. Well, that's what your cell will be trying to do. If it's not in its interest to keep a certain gene, it will try to mutate it out. And this evolution can totally break your biological system. So we are working on a bunch of challenges, and it's actually systems theory and mathematical modeling and disciplines like this that allow us to deal with this subject. So that was rather exhausting consideration and dispute on synthetic biology. And now let's delve into diagnostic and therapeutic applications of synthetic biology for the next around 20 minutes, and then we can open it up in a discussion. So basically, we'll be talking about about understanding of fundamental engineering principles applied to a couple of different topics, including vaccines, paper-based Zika tests, CRISPR-based SARS-CoV-2 tests, engineered CAR-T therapy, and engineered bacterial and cellular therapeutics. And what I put into the brackets here are basically either the companies or the institutions that are bringing these innovations either to the market or to the people, because I just think that it's very important to give the credit where the credit belongs. So synthetic biology enabled vaccines. So what are the fundamentals of the vaccines? So on a very fundamental level, we need to select an antigen and get the antigen to the body to induce the immune response and induce the non-specific and specific immunity. And historically, we know that attenuated viruses or dead viruses or viral fragments have been used to do that. Our synthetic biology offers three different ways how to deal with vaccines. It's codon-deoptimized vaccines, DNA vaccines, and mRNA vaccines. So in kind of codon-deoptimized vaccines, let me skip a slide forward, is that basically we take the genome, or like we take the pathogen of interest, sequence it, so read its genome, and then kind of de-optimize the sequences of this genome either manually or using computer algorithm. So and why do we do it? Well, we do it to like really attenuate, say, the reproduction ability of this virus. So for example, as you know, there is something called codon redundancy. So there is more codon triplets than amino acids. So there can be multiple codons that go for the same amino acid. However, there are different codons have different presence or abundance in the organism. So we can kind of make it much more difficult for the virus to replicate if we introduce less abundant codons. So we can also de-optimize codon pairs, and as other thing is that we can do basically substitutions of the codons that will become somehow less favorable, that will allow say single mutations to give nonsense codons. And so then we like go and synthesize these de-optimized sequences. We generate the full length of like coding DNA, incorporating these de-optimized coding sequences. We recover the viruses with de-optimized codon sequences by kind of reverse genetic, evaluate them for attenuation in vitro and in vivo. And it can be like, oh, like why the hell, like would you go with this? Because like what this actually enables us to do is to grab the unknown virus, like break the unknown virus, make it like really unoptimized and inefficient, and make a vaccine out of it without like having the great, without having the great detailed biological knowledge. So, and we can do this very rapidly, often. So, and this gives us immunity similar to natural infection because it's still the same virus, and it's very rapid generation. So let's imagine maybe in the future we have like some crazy pathogen like zombie, literally like zombie virus. Zombie virus comes, like we take the virus, sequence it, like break it, give it to people, and get the immunity against it. So it would be quite, and we can imagine that it could be very difficult for the natural virus to like somehow get around this, because like if you introduce that many mutations, like the natural virus might not be able to get away with it. So there are obvious disadvantages, like different viruses require different de-optimization. This can still cause the illness in immune compromised people and might require a viral culture. Then we basically have these DNA vaccines, which I will just talk briefly about because they haven't been used in practice. I mean, codon-deoptimized vaccines are still in development. I think this CODAVAX is quite interesting against, this is for codon-deoptimized against universal influenza and against SARS-CoV-2. This could be quite a nice proof of concept, but the other are still in kind of either GMP manufacturing phase or even in vivo animal studies. But DNA vaccines are very interesting because they can somehow they have high temperature stability, right? So you don't have to worry with the Pfizer at -70, have rapid design and manufacturing, but have the potential for genomic integration. So we certainly don't want your DNA of a, so say, reversely transcribed DNA of a virus to be included in your genome. And this requires a lot of biological knowledge for target selection. So RNA vaccines also have been used. We know about Moderna, we know about Pfizer. They have tailored epitope presentation and the residence time. They can basically self-amplify and are rapid design and manufacture. So we can basically go design the sequence, the mRNA of the specific viral segment that is kind of non-infectious, and we can encapsulate it in lipid nanoparticle and let our cellular machinery to produce this protein, which then induces the immune response. Well, what are the disadvantages? Well, this requires multiple doses and again requires a serious biological knowledge for target selection. So here we basically can see how mRNA vaccines work. Like, I'm sure that I don't have to explain this to the medics. What is very interesting about mRNA vaccines is that mRNA vaccines can be used not only against, say, infectious diseases, but a lot of work is being done on mRNA vaccines against cancer, or a lot of mRNA vaccines. Now, currently, an mRNA vaccine is being developed against multiple sclerosis that has shown some remarkable efficacy in animal studies. So there is definitely the path forward. And what is remarkable is that Jonas Salk, it took at the order of 20 years to develop the vaccine against polio. Well, it's taken about 1 year to get a COVID-19 mRNA vaccine. It's not that the idea was there, obviously, and Moderna, for example, and BioNTech have been working on mRNA vaccines for some time. But kind of it's so remarkable that from the inception of the pandemic, so from the discovery of the virus up to the point where we got to the vaccine, to the functioning vaccine, it took us around a year. Another interesting application, I believe, is the paper-based Zika test. So Zika can be passed from a pregnant woman to fetus and caused birth defects. It was a huge pandemic, I believe, in Brazil in around 2015, 2016 or so. There's no vaccine or medicine, and in that outbreak, around 5 years ago, I believe, there were around 200,000 cases with 8,600 birth defects. And what was required was a rapid, low-cost, and reliable test against Zika, especially given that typical testing often faced false positive with dengue fever. And synthetic biologists mostly from the MIT and Harvard used a toehold switch sensor, which is basically a piece of RNA, which after the binding of the target RNA, so some external RNA, right, the target RNA, and the picture at right, undergoes strand displacement, allows for this ribosome binding and translation of the protein, which if it's lighting protein, if it's fluorescent protein, can produce the visual output, or if it's a colorimetric assay. So what you can do is to freeze dry this in the paper and get a paper-based diagnostic. So Zika tests were quite difficult to do in local conditions. So again, these researchers combined two things: programmable RNA sensors and freeze-dried paper-based cell-free protein expression platform. And literally, they optimized their development process so fast that it allowed them to, develop the sensors in around 5 days. Then you get them to distribute and store at around 1 year at 20 degrees, which is remarkable. Like not -20, not -70, 20 degrees. And then you could do the diagnostic test from sample collection, RNA extraction, RNA amplification, and toehold reaction with visual detection in around 3 hours. So this is truly remarkable. Synthetic biology and design allowed us just in a couple of days, again, at the order of days, to go and develop the diagnostic test against this dangerous pathogen. And similarly, I think that all of us are aware of like current SARS-CoV-2 pandemic, and especially in our countries, in Slovakia or Czech Republic, there was a lot of demand for the testing. So there was in 2020, there was this new method presented called Sherlock, which is specific high sensitivity enzymatic reporter unlocking. And a weird that is a weird abbreviation, funny one though, that combines a couple of techniques that I'm not going to get into detail of, that allow to do a very rapid SARS-CoV-2 testing. So at a very simple, so again, what do we want to with the SARS-CoV-2? We want to diagnose or detect the presence of the target RNA. And basically with enzyme called Cas13, so usually people talk about CRISPR-Cas9, but here we have CRISPR-Cas13a and CR-RNA. We can, and target RNA, where we can get to is that we can basically break the bonds between biotin and 6-FAN, and then get the binding of the antibodies to the complex, and basically get a typical kind of lateral assay tests. So and similar as we can see with this kind of antigen testing. However, this test, if you think about it, has kind of sensitivity comparable to RT-qPCR, can be done at a single temperature in less than an hour and less than $10 per test, and it's still like very easy to do. So again, let's imagine that you have your PCR not in 2 hours and with the expensive thermocycler, but in less than an hour, less than $10 per test at a single temperature. And that is, again, that is very, very remarkable. So after the picture, you can see kind of the protocol, which is kind of, very simple, very simple. And you can basically also see the flow of the test. So we briefly went through the diagnostics, and we have shown we went through the vaccines, so prophylactic, diagnostic. But now let's talk about major challenges in treating challenging diseases. So just yesterday, I went to the talk by Professor Tim Liu from MIT, where he pinpointed 4 difficult kind of things about complex diseases, which are dynamic conditions of the disease that make it difficult to pinpoint the disease, lack of specificity of the expressed target in the disease. So often our drug would, even though meant to be specific, will kind of have different off-target effects. Narrow dose window of therapeutics. Like, so what you can do, like you either take the tablet or you don't take the tablet often, and that's that's not like the best adjustable dosing. And also you have kind of very narrow window where your therapeutic might be effective, and also disease evasion of the therapies, even the specific one. So what would we like to have as a treatment to diseases? We'd like to have topical, so that means kind of local, reactive, low cost, and safe medicines. And engineered cellular therapeutics can actually hopefully provide that. In what many of us are hoping in is that we will basically be able to have the cells that can sense the external regulatory signals or cellular disease biomarkers or systemic disease biomarkers, then contain the logic processing, this kind of genetic circuit, as I mentioned, that can say, evaluate the combination of these different biomarkers and come and say, okay, biomarker 1 and 2 is present, biomarker 3 is not present. It is kind of, I should make this. And that can be regulated therapeutic function, which can be altered gene expression or change in protein activity or some metabolic activity or secretion. And there should be some kind of feedback regulation, which tells me that somehow I have produced enough of this, I should stop. And this is what we want to do with engineered cellular therapeutics. And this is, for example, what is being done with engineered CAR-T therapy. So in CAR-T therapy, which is one of the cutting-edge treatments for, say, liquid tumors or liquid cancers, is that we remove blood from the patients to get the T cells. We make the CAR-T cells, which means CAR means chimeric antigen receptors, where we insert this chimeric antigen receptor in the membrane of the T cell. We grow millions of them and infuse them into the patient, and then hopefully CAR-T cells binds to the cancer cells and kill them. Well, what is the issue with that? One of the issues basically is that we would really like to have a very specific binding of these CAR-T cells to these cancer cells, and that's one thing. But there's other thing. We should somehow very tightly regulate, say, how these T cells, how strongly they kill the cells, to put it in an abstract way. And this is where the synthetic biology and again this complexity comes in. Perhaps we would like to be able to only our CAR-T cells, say, even express some of these chimeric antigen receptors at a certain level and at a certain time, say when present next to the cancer cells, in the presence of the cancer cells. And this is what companies like Centibio are working on. It's basically using the eukaryotic cells, T cells, natural killer cells, iPSCs, to basically engineer them to allow them to produce useful therapeutic compounds and to, say, regulate their effect so that we can achieve the better therapeutic outcomes. And finally, let's continue with this engineered cellular therapeutics. So on the left, what we can see is a truly remarkable work of Martin Fusanegra's lab, where they basically constructed the eukaryotic cells that could be administered systemically to the mice that were able to detect the levels of TNF, I think it was TNF alpha and interleukin 22. And if these both of these biomarkers were present in increased amount, these cells were able to produce interleukin 4 and interleukin 10, which basically allow them to help address the psoriasis. So this was the engineered cellular therapeutics for psoriasis. We've got engineered cells, engineered bacteria by a company called Synlogic against the phenylketonuria, which is basically the condition in which patients can't metabolize phenylalanine, and this company basically makes bacteria, develops bacteria that would be inserted in the gut and would contain the enzymes for the metabolization of phenylalanine into basically hippuric acid that could be basically excreted in urine. So this is basically the work of Synlogic. So instead of patients with phenylketonuria, like having severe dietary restriction, which is still like very difficult to obey, you could simply have bacteria in your gut that would be able to eat and metabolize these compounds. And also in the cancer, like one of the novel elements where people are looking at is cancer and complex bacterial therapeutics for cancer. So what we know is that some bacteria can localize to the tumors, and hopefully in the tumors we can have the inducible promoters that can be activated by the local hypoxic environment and kind of produce therapies on demand directly locally in the tumors. So say I believe that there's some research by Salmonella typhimurium that was able to localize into the tumor cells and produce and basically produce therapeutics there. And the other interesting application is synthetic virology, where we can recombinantly produce different viruses to study them better and to basically study their genetic modification for good purposes. And always people think about biosafety, and I think that biosafety is very important, and not even the biosafety related to synthetic biology. I mean, the fact that 6 vials of smallpox were discovered in a US lab has nothing to do with synthetic biology, but rather with people being careful on like what box where you put. But I have a couple of slides on like how we can make sure that our engineered bacteria are safe. And considering the time, I've been talking for around an hour, I will kind of skip over these, and I'm happy to elaborate on them if you like in the discussion. And I will also just say that we are now trying to build the cells from scratch, and there are kind of two. There's really a great initiative called Build a Cell, which aims to build a cell from really de novo, from essentially synthesized chemicals. And there is also work of J. Craig Venter that was able to synthesize the genome of the smallest existing living cell, which is unnatural, called JCDIC 3.0. And that is very exciting research, and synthetic cells have a lot of implications for therapeutics. So in summary, synthetic biology is the engineering discipline that aims to design and construct novel biological systems and living organisms. It has a potential to transform diagnostics and therapeutics, as we have seen. We need to focus on safety, regulatory matters, and scaling of the production of these useful things, because it's one thing to do something at a bench, at the lab bench, and it's different to scale it up to the industrial level, as we can see with the vaccines effort. And we need better interplay between clinicians and bioengineers to build better therapies and diagnostic tools. And I just want to say that synthetic biology is exotic, as you can see on that engineered pineapple at the right, economic, as you can see on the left, in this graph of decreasing cost of sequencing per genome, and above all is beautiful, as you can see on this Mona Lisa picture at the lower left, which is actually printed into the bacteria. And with that, I'd like to thank you for your attention. Hope you enjoyed the presentation, and I'm happy to engage in a discussion now.